researcher and consultant. She will be speaking on aphids, and it's particularly good that she's with us today because she's just tested negative and just recovering from COVID. So she's off her sick bed to present to us this afternoon. Then Jude Bennison, who I'm sure you will all know, entomologist, will be talking to us about thrips in detail. And then finally, last but not least, David Talbot, ADAS consultant, will be talking to us about whitefly. So without further ado, Alicia, would you like to start?
Right, hello everyone, thanks Chris. You'll have to bear with me a bit as I'm still getting over COVID, but off we go. So we're talking about the big three pests today that I'm sure you'll all be familiar with, and you should be able to recognise which pest caused the damage that you can see here and answer the poll that should have popped up on your screen. And I'll just give you a few more seconds to answer that poll. And Coral, can you send me the results now? Yeah, 100% that I got aphids, and so that's what we're talking about today. Aphids are a big problem because they suck plant sap to obtain sugars and proteins. They reduce plant figure and cause distortion such as twisted or curled leaves, yellowing foliage, and they slow down plant growth. They also excrete honeydew, which leads to the growth of sooty molds, which reduces plant quality. And some aphid species consume this honeydew and will defend aphid colonies from their enemies. And aphids are also well known for transmitting viruses. But have you ever thought about what makes aphids such a bad pest? It's all about their life cycle and how rapidly they can reproduce, which you'll get an idea of on this diagram here. At stage 1, the aphids develop through 4 larval instars, and at stage 2, the adult females don't need any males and they can reproduce live young by parthenogenesis. And at stage 3 is the production of a winged form, which is known as elates. And these are usually produced when the colony is overcrowded or the host quality is poor, so that they can spread to a new plant. And at stage 5, most species have both a summer and a winter host plant, and they perform sexual forms during the winter part, which produce eggs for overwintering. However, in temperate climates and in bedding nurseries particularly, which are nice and warm, they'll produce their live young all year round. So some species of aphids are found on specific hosts, but the main culprits that you'll come across are the prolificous species, which can be found on many hosts. And the main 4 are Mysis pesci, Aphis cassipii, Macrosiphon euphorbia, and Olacorthum solani, which are found commonly on the plants shown here. It's very important to know which aphid species you have, as different species require different control options. For example, Mysis and Aphis cassipii are resistant to several insecticides. But how are you supposed to identify these aphids? There are some helpful guides like the Bayer Expert Guide and this BioBiz poster, but to be honest, aphid identification is hard, even with a microscope and morphological key. And I wouldn't even try to bothering to identify the winged aphids. But there are some basic recognition features that will help you decide which aphids you have. And if you're not sure, you can always send a sample to us at ADAS or to another expert. The main features you'll be looking at are the abdomen size and shape, the antennae length and markings, the shape of the antennal tubercles, which are the part of the head the antennae are attached to, the siphuncuли, which are the exhaust pipes at the back end of the aphid, and they're used for releasing pheromones, and the cauda, which is like a tail, and it's used for flicking droplets of honeydew away from the aphid. And you'll be surprised at how many of these features you can see with a good hand lens. But don't pay too much attention to colour, as different species have many different colour morphs, which I'll go into now. Starting with Mysis pesci, the peach potato aphid. It can be green, pink, or red. It's a very small aphid, 1 to 1.2 millimeters long. The antennae are roughly the same length as the body, or a bit shorter. The siphuncuли are medium length with slightly swollen dark tips, and the adults have a short cauda, but this can't be seen on the nymphs. And the gap between the antennal tubercles makes the W shape. It's typically found, as you can see here, on the underside of leaves, on growing points and stems. And I found out during an experiment that they really like pansy flowers too. And they can also be found in dense colonies quite low down in the plant. And Mysis pesci is the most important pest and virus vector aphid in Britain. Aphis cassifii, the melanin cotton aphid, is a prolific species with around 900 host plant species. And despite this large range, it tends to reproduce on its chosen host all the year round without switching between species. It's a very small aphid with a pear-shaped body, sometimes reminds me of a mite. The body is around 0.9 to 1.8 millimeters long, and size can vary with plant host and host quality. I've worked with some very tiny Aphis cassifii. The antennae are generally shorter than the body, and the siphunculæ are short and black. The cauda is short and paler than the siphunculæ. And this aphid is quite easy to mistake for Aphis fabi, the black-green aphid, which is black with a dark cauda. And the colour of the Aphis cassifii can vary between a blackish green to a very pale yellow. And often it's in hot, crowded conditions which cause the colour to change from black to yellow. And this aphid particularly loves stems and flower buds in my experience. And you should also be aware that I often find it in mixed colonies, both mixed by colour or sometimes mixed with other species. As you can see in the bottom right, here it is with Mysis pesci. Macrosiphon euphorbiae, the potato aphid, has over 200 plant hosts but prefers solanaceous species. It's a large aphid, that's around 1.7 to 3.6 millimeters long, and often has a dark stripe down the center of its back. The antennae are longer than the body, and it has particularly long legs and siphunculæ, and a finger-shaped cauda. It has an elongated body, and colour varies from light green, yellowish green to pinkish red. Aureus solani, known as the glasshouse potato or the foxglove aphid, feeds on around 200 species as well, and particularly affects a range of houseplants. It's another large aphid, 1.5 to 3 millimeters long, with antennae slightly longer than the body, with dark joints found on it. It has long siphunculæ with black flared tips and has dark patches at the base of the siphunculæ. The gap between the antennal tubercles makes a U shape as well. It has a pear-shaped body, and it could be whitish green or yellow, dull green or greenish brown. So how are you supposed to control these aphids? I hope you're already using integrated pest management, which involves following this basic pyramid of principles that I'll go into now. Starting with cultural control. Aphids love weeds, particularly as winter hosts. So when you go into the glasshouse, always look for these particular weed species. That's chickweed, groundsel, and sow thistle. Check them for aphids and then remove them. I won't go into how to control weeds, as that's a topic for another webinar. Severe aphid infestations can be a source of winged aphids, which will spread around the nursery. So often it can be better to bag them up and remove them rather than try to control them. Aphids also feed on plant sugar and love new soft growth. So if excess fertilizer is used, then there'll be a lot of soft growth and sap to feed the aphids and allow them to reproduce even faster. So looking at encouraging natural enemies, probably the most famous of them all is the ladybird. And although it's important to remove weeds from within the glasshouse, a lot of research is showing that maintaining some semi-natural habitat outside the glasshouse provides overwintering sites for natural enemies such as ladybirds. Nettles are a particularly good host for beneficials, as they've been found to support around 100 species, and they usually get the nettle aphid, which will not transfer to your crops, but helps to boost numbers of predators. Reducing use of pesticides will also help to encourage natural enemies, and most importantly, be aware of what they look like. So here you can see the ladybird larva, the pupa, and the adult, and there are many different species in Britain, all looking slightly different, with different larvae and pupae too. The ladybirds are great. I was amazed when I left this campus outside, and within hours it was covered in ladybirds, and the next day it was picked clean of aphids. But in general, ladybirds, particularly the adults, are not to be relied on for aphid control, but they can be bought commercially if you're in a pinch. Hoverflies are an even more voracious predator of aphids, and again there are many different species, and all the larvae and pupae and adults look slightly different. Hoverfly larvae can eat enormous numbers of aphids, but the adults need access to pollen and nectar to feed on. They also need large number of aphids to support their young, so you tend to see them after your aphid infestation is already out of control. But some growers are seeing the benefit of releasing these predators commercially. And also just be aware here of the hoverfly parasitoid Diplazon and later Aureus, which is something you might see on your nursery. Lacewings are also terrific predators because they are generalists and they'll eat many other pest species too. In fact, they're so aggressive that they can even bite humans. They are also commercially available and they used to be quite expensive, but they are coming down in price now. A lesser known natural enemy of aphids is the predatory mite Anastas Bakarum, which I witnessed feeding on Aphids cassipi in the picture on the left. And there was a campaign in Northern Ireland to stop farmers from spraying for this mite as they had it confused with the fruit tree red spider mite. So it's good to be aware of it. And on the right, what looks like bulging eyes is actually the alarm pheromone being released from the siphunculate of an attacked aphid. Another thing to be aware of is the natural entomopathogenic fungus Pandora Neophytus, which forms an orange-brown mass on the dead aphids. If you see this, that's what's going on, but it likes humid conditions. So if you are seeing this, your relative humidity may be too high. So you should be looking out for all these things during regular crop walks, and the best tool for monitoring aphids is a hand lens to look for the aphids when they're in low numbers, particularly by searching the underside of leaves, growing points, flower buds, and the calyxes. Ideally, you'll find them before you see the symptoms of distorted growth, cast skins, and sooty mold. There's an AHDV forecasting system for aphid species focused on cereal crops, but the Mises persicae forecast can help to inform you when Mises numbers are high and likely to migrate from nearby fields. There are also several apps being developed at the moment. Anastas is working on an app to identify aphids in wheat from photographs, and apparently it's very good. So you're already monitoring using decision support and probably have a history of aphids. So it's wise to preventatively release parasitoid wasps, even if you can't see the aphids, as they'll be able to find them before you can. And these can be released weekly, and rates increase when the aphids are seen. Parasitoid wasps lay their eggs inside living aphids, which then hatch into the next generation. So this is when it becomes important to know which aphid species you have, as some parasitoids are better for parasitizing some aphids. Aphidius comani and Aphidius matricaria parasitize the small aphid species, that's Mises and Aphids cassipi. In general, Aphidius comani is better than matricaria, but matricaria covers a wider host range. And Aphidius urvi and Aphilinus abdominalis parasitize the big aphids. And of these two, Aphidius urvi is the better choice, usually. However, most biocontrol companies sell parasitoids in mixes. So if you don't know which species you have, or it's not one of the main four, it's probably better to buy a mix. If you do know which species you have, you could save money and get better results with a specific species. As the parasitoid larva develops inside the aphid, it forms what we call a mummy. And here you can see a picture of a parasitoid developing inside an aphid before it turns into this characteristic golden brown color associated with the Aphidius species of parasitoids. Aphilinus abdominalis, however, forms a black aphid mummy, and Praeon volucre forms a distinct mummy on a white pedestal. And once the parasitoids have left the aphid mummy, we get more useful information from the shape of the exit hole. A parasitoid will leave a round exit hole, whereas a hyperparasitoid, which lays its eggs inside the developing parasitoid, inside an aphid mummy, will leave a jagged hole. And if you rely too heavily on parasitoids, you can end up with a large population of hyperparasitoids, which go around killing your purchased biocontrols. To reduce build-up of hyperparasitoids, it's best to incorporate predators into an IPM program, and the predatory midge larva Aphids aphidomizer is a great choice. And I know this because I have lost a lot of aphid cultures to this midge. It can be released once daylight hours exceed 15, and that's between May and September, and it performs best between 20 and 30 degrees. It particularly needs it to be warm at night when that's when it's active. Be aware that high numbers of generalist predatory mites may feed on its eggs, which are bright red if they're short of prey. If aphids are an issue, ladybirds, hoverflies, and the green lacewing can all be commercially purchased, and as of recently, so can the brown lacewing. It will also predate other soft-bodied insects, and it's suitable down to temperatures of around 10 degrees C. Both the adults and larvae can consume aphids, but it can be cannibalistic, so best not used preventatively. Moving on to sprayable options, there's the entomopathogenic fungus Bavaria bassiana, which is a fungus that occurs naturally in soil. Its spores penetrate the cuticle of the host and kill it. The products Naturalis L and Botanica WP are approved for whitefly and thrips control, not aphids. But in the AHDB Septoplus project, I completed a review with Jude Benison on independent efficacy work done on aphids, and I'll share some of the outcomes here. And trial work has shown that Bavaria can be used to reduce numbers of mysis and Aphis cassiopeiae when tank mixed with majestic. The neem seed extract azadirachtin is the active ingredient in azatin, which is approved for thrips control and also has no claim against aphids. However, in the AHDB funded AMBER project, which aims to improve performance via pesticides, we developed a model for predicting the efficacy of azadirachtin against mysis pesceae. And you can see the results here, showing that one spray would not be very effective, but after 4 sprays, the population should be eradicated. We set up a trial to validate this model on Pansy in 2020, and we'll be talking about the results of this in a specific AMBER webinar in June. Other trials, however, have found that azatin against aphids found no significant reduction, but they were using a lower rate than is permitted on ornamentals. And this table shows IPM compatible products for the control of aphids. And I've determined IPM compatibility by checking the BioBest and Copper side effects guides. And these products tend to have less than 50% mortality on aphid beneficials, or they only have a very short-lived effect. I used Eradicote Max in a Septoplus trial in 2020 and saw a significant reduction of mysis pesceae, but not Aphis cassiopeiae. Aphis cassiopeiae is very difficult to target with contact-acting products as it's protected by a waxy layer, and because of its small size, it hides very easily within plant crevices out of reach of sprays. Other trials have shown no significant reduction by majestic, but it was effective when tank mixed with Botanigard, and it will be replaced by Eradicote Max, which is not yet available but has improved efficacy against aphids. Products such as Mayman and Batavia with EMO approval are only to be used at the grower's own risk. And SB Plant Invigorator and Protac SF are exempt from registration as they're not approved as pesticides, but one study found a 49% reduction in mysis with SB Plant Invigorator. These products should be used with care as they can cause harm to beneficials. That's more than 50% mortality. They can be used as spot sprays or for a quick knockdown or cleanup prior to dispatch. Systemic pesticides are best used when weather is warm so that the plant can actively take them up, but can be very effective in crops with a dense canopy where good spray coverage is tricky. Gazelle is a neonicotinoid and Sequoia is considered a 5th generation neonic, and some retailers don't want these products used anymore. Sequoia, however, has performed very well against aphids in a number of studies, but it has an 8-day handling limit after application. Products like Sprutzit only have a short-term effect on beneficials, but Dacis forte and Hallmark can leave harmful residues for a long time, which might only be suitable for use in winter when beneficials aren't active. You should check the BioBest side effects manual and the COPET side effects database to better understand how products affect your biocontrol program. You'll notice that very little efficacy work has been done on bedding plant species, as it's a small sector, but we can gain a lot of transferable knowledge from the other sectors. As there aren't many pesticide options available, and most have a limited number of applications, this is to encourage growers to switch between mode of action groups classified by the Insecticide Resistant Action Committee, known as IRAC. Aphids are prone to developing resistance, and Mysis persicii and Aphis scicipii have developed resistance to carbamates, organophosphates, and pyrethroids. And outside of the UK, there's evidence of resistance to neonicotinoids. It's also possible that aphids with the target site mutation for resistance to neonics could have cross-resistance to similar chemistry like Ciguel. And if you've been noticing reduced efficacy or suspect you may have a resistant population, please send samples of the main 4 aphid species to me at ADAS Boxworth, as HSE have recently commissioned ADAS to do baseline sensitivity testing on the 4 main aphid species, particularly in protected crops, which is great as these don't always get a lot of attention. We'll be doing the resistance tests ourselves, so we will be able to give you feedback on the results. And if you email me, I can send you prepaid envelopes for posting your aphid samples. Now, I know aphids have been covered in many other webinars, so I'm hoping that you might have learned something you hadn't heard before. And a poll should be popping up now, if you can answer that. Just give you a few seconds to answer while I catch my breath. Great. Coral, could you bring the results up, please? Great. Oh, that's a, I don't know if you can see those results or not, but 93% said yes.
We can, Alicia, yeah.
Great. Okay, now if there's any time, are there any questions?
Right. Well, just right. So we had a very interesting question from Sam Brook and a very good answer from Andrew Gladman. So I'd recommend, we're quite short of time, so I'd recommend everyone just have a quick look at the chat. And it was a question that we asked whilst Alicia was doing her practice run a day or so ago. So I'd recommend everyone goes and have a look in the chat there. But if we'd like to move on now, and if Alicia would like to stop sharing her screen and Jude would like to start sharing hers, we'll move on to thrips. Excellent. Okay, all yours, Jude. Jude, I don't think we can hear you. You're on mute, Jude. You're on mute.
Okay, can you hear me now?
We certainly can. That's great.
Good. Sorry about that. Yeah, okay, so I'm sure you all know who caused this damage to the flowers and leaves. So I'll give you a little bit of time to do the poll. Okay, Coral, have we got the answer? 96% thrips, 4% whitefly. Okay, well, it is actually all done by thrips. A few of you might have thought these were sooty molds caused by whitefly, but when you get thrips feeding damage, you always get little black specks, which are the droppings that they leave behind when they're feeding. And for those of you who are not familiar with this, this is a symptom of tomato spotted wilt virus on impatiens that is transmitted, can be transmitted by Western flower thrips. So well done, everybody. So here we have our old friend Western flower thrips, which needs no introduction. I think it's important for you all to know the life stages of the thrips, because the different life stages can be controlled by different biocontrols or plant protection products. So here we have the female, and the female is slightly larger than the male. Here you can see them on sticky traps. The male is smaller than the female and yellow, whereas the female is yellow at the front and darker at the hind end. She lays her eggs in the young plant growth, which hatch out into these first instar larvae, which are very small and pale. They develop into the yellow, larger second instar larvae. And when these are fully fed, they usually drop to the ground to pupate. And you can see the pre-pupa and the pupa here. Most of you probably never seen these pupae because normally they're in the ground, although sometimes you can see them on sheltered parts of the plant. And when the pupa is ready to emerge, they emerge as an adult, which goes off and finds another of your plants to lay eggs in. Can you all see the full screen without these? Oh yeah, it's me. I had all the sort of images of everybody at the side, so I've got rid of those now. Okay, so these are just some of the species you can find on bedding and pot plants. There are more, but these are the ones I thought I'd show you. So we've got Western flower thrips, we've got onion thrips, Thrips tobacci, which looks quite similar to Western flower thrips really, and can be found on a lot of protected crops, although Western flower thrips is more common. And then a couple of years ago, we started having problems with this thrips, the Japanese flower thrips, Thriptosis, which I'll speak a little bit more about shortly. It has these very characteristic white wing bases. I always think they look like they have white shoulders, but that's not necessarily means it's Japanese flower thrips, because you can see this one and other species also have these white shoulders. This one is so-called Vanda thrips, Dichroma thrips corbeti, but that one's specific to Phalaenopsis. So unless you're growing them, you won't be seeing those. Wayne Brough from AHDB asked Herben Messelink at Wageningen to translate his Dutch poster on thrips recognition into English. So that is available. If you haven't got a copy already, it's available on the AHDB website. So a little bit about Thriptosis then. It came into the UK in 2016 on imported plant material. The first case was confirmed on poinsettia, but after that we had most problems on cyclamen, New Guinea impatiens and herbs, particularly basil. In other countries, it has a wide host range on many other ornamentals, bedding and pot plants, nursery stock and cut flowers. It'll also go on protected and outdoor edible crops and weeds. So yeah, it could have potential for spreading into other crops. It is more of a leaf feeder than Western flower thrips, but it will also go in flowers. You can see them here on this white cyclamen flower. And it does cause more damage than Western flower thrips to the leaves. This is the underside of a cyclamen leaf. And as you probably all know, normally with Western flower thrips, you don't know you've got it till the flowers start to open. But with Thriptosis, you get this very evident scarring. This is a little movie that James Lisher from Cobbins Nursery very kindly sent me. These are Thriptosis females wandering about on a cyclamen leaf. You can see him prodding them there with the end of a pen. If that had been Western flower thrips, it probably would have flown off by now. And that also indicates that maybe sticky traps aren't as good for monitoring this one. I don't think it's as an active flyer as Western flower thrips. So that's a bit about recognition of thrips that you can do. To actually identify them to species, you need to send them to somebody like us, because we need to mount them on glass slides, look at them under high power, and look at small features such as number of antennal segments. So this is Western flower thrips. It has 8 antennal segments. The end one is split into 2 very small ones. This is the antennal segment of Thrips tobacci, the onion thrips, and that only has 7, like most other thrips species. The end one is not split into 2. With Western flower thrips, these are the 2 compound eyes here, and they have this whacking grey hair behind the eye that sticks out. Whereas with thrip species, such as Thrips tobacci, the onion thrips, the hairs are all quite short behind the eye. So we use quite a lot of other features to identify them to species. Not all thrips are bad. This one's actually a goodie. It's a predatory thrips, Aeola thrips intermedius, which actually feeds on other species of pest thrips. So You don't want to be killing that one off. It's bigger than pest species of thrips. Here you can see them mounted on a microscope slide. You can see they're twice the size of this pest species here. It has these nice rugby striped wings. So if you see that, that's good. And we often see them in strawberry crops where people aren't using many pesticides. I haven't seen them that many in ornamentals, but maybe you've seen them. So you all know about IPM is a combination of cultural control, monitoring, biological control, and use of compatible bioprotectants or conventional pesticides. I updated the AHDB fact sheet on thrips control on protected ornamentals last year. Here's the link. So anything I haven't got time to talk about today, if you just press on that, it'll take you straight to it. So cultural control, as Alicia says, weed control is very important. Groundsel, as well as being a host for aphids, it's also very commonly infested with thrips. And you all know that nursery hygiene and disposal of old or infested plants, and also the matting and any plant debris, because they could harbour the ground dwelling stages. So careful disposal is very important. You can use these long roller traps. For example, if you've got an old crop of bedding that's got thrips on and you're about to take the leftover ones out at the end of the season and you want to stop them flying to your new cyclamen crop, you could put these up in between the two areas. So monitoring with the regular sized thrips, I'm sure you're all using these. But don't forget to check the plants. Don't just rely on the sticky traps. So check the plants on arrival and throughout the production period every week. You can also use indicator plants. For example, if I was walking into a glass house full of bedding plants, I might home in on verbenas because they're very susceptible to thrips damage. It shows up very easily on the flowers. For tomato spotted wilt virus, petunia or impatiens, they show the symptoms up very quickly. In general, blue is best for Western flower thrips. However, in low light conditions in the winter, yellow can actually be better. And of course, they're easier to see on yellow. And reminder here of the female and the male Western flower thrips. But as I mentioned, Thriptosis isn't as an active a flyer as Western flower thrips. It has been caught on sticky traps on nurseries, but I know in the Netherlands, they've told me they don't bother with traps for Thriptosis monitoring. They prefer to just focus on the plants. Those of you who listened to the BPOA webinar last January when Claire Sampson from Russell IPM joined me to talk about thrips trapping, and most of you will know that there is a specific Western flower thrip sex pheromone which comes as a little rubber thing that you stick onto your trap. It actually attracts both females and males, and it can double your trap catches. So that might be useful if you're trying to detect the first thrips when the thrips are at low densities. But that only catches Western flower thrips. We're currently working on an AHDB project on strawberry where they have a mix of thrip species that can damage strawberry, including Western flower thrips and onion thrips, but also a couple of other species. And last year we did a trial and we looked at the copper laurum, which is not a sex pheromone, it's actually a plant extract which is attractive to various thrip species. And you can see here we found 3 times as many thrips on the laurum traps as on the untreated traps. And Russell IPM are developing have developed a new thrips law called Thrip Knock, which is commercially available, but we found that in strawberry it wasn't as effective as laurum, but was slightly better than the untreated. So we're continuing with that work this year, and it might come up with some more results that might be useful to you ornamental growers as well. So over to biological control, Neosulus cocumurus, the predatory mite. Most of you will be using this as the basis of your IPM program. It's cheap, it's got a wide temperature range, it's got no restrictions on release because it's native to the UK. It only feeds on the first instar or the first stage larvae of Western flower thrips. It doesn't eat the adults or the later stage larvae. However, it can be very effective if you start it preventively and put it in every week. But there are some other predatory mite species now available for thrips control: Montdorensis, Limonicus, and Swirsky. They all have slightly different temperature ranges. Limonicus is more of a cool temperature beast, whereas Swirsky likes it warm. Now, all these species will eat both the first and the second stage larvae. You can see one having a go at a second stage larva there. So they are actually better predators than Cocumurus if the temperatures are okay. And they'll also feed on whitefly eggs and young scales. So if you've got plants that have got thrips and whitefly, they can be helpful there. But none of them are native to the UK, so they can only be released under protection. And they are more expensive than Cocumurus, but because they're better predators, you can get away with lower release rates sometimes. So as well as predatory mites against the thrips larvae, we also have predatory mites that are ground dwelling and feed on the thrips pupae. This one is Hypoaspi, which has actually been renamed now, Stratiolelaps, but it's still mainly sold under the name Hypoaspi. Then we have our entomopathogenic nematodes that are applied as a foliar spray, but actually their main action is in the growing media against the pupal stages. And you can see a pupa that we've dissected here in a drop of water and the nematode floating out. You only need one nematode to get in a thrips to kill it. Then we've got our predatory bugs, Aureus, which are good because they'll feed on thrips adults as well as larvae. We have our entomopathogenic fungi, Botanigard, Naturalis, and Mycotal. You can see the fungus growing on the body here. And Aphita, which again has been renamed Delosia, but many of you will be using this perhaps for Sierrid and shorefly control. It will also have a go at the ground dwelling stages of the thrips. There's various release methods for predatory mites. Some of you will be releasing the loose method, some of you will be using sachets, or there's the automated devices such as the Coppet airbug. Overseas, they're now beginning to use drones to release predatory mites and other biocontrols on outdoor crops, and I'm sure before long they'll be starting to be used over here, even in protected crops. This is just to show you how good Cocumurus can be, not only for controlling damage, but also controlling transmission of tomato spotted wilt virus by western flower thrips. This was in a trial we did some years ago. This is a plant where we'd released Cocumurus. This is a plant from our untreated control, which had been hammered by the virus. So it just shows you how good Cocumurus can be. However, it doesn't seem to be very good against Thriptosis, because when this came in, the growers who had Thriptosis were all using Cocumurus, which was giving very nice control of their western flower thrips, but it wasn't controlling Cytosis. So they all had to start using pesticides, which of course disrupted their IPM. So AHDB asked Alicia and I to do a review on the biology and control of Cytosis. Here's the link here so you can get further details. As part of this review, I contacted someone I know at Coppet Research in the Netherlands, and they'd just done some work in the lab that showed Montdorensis, Limonicus, and Swirsky will all feed on both the first and second stage larvae of Cytosis. They hadn't actually tested Cocumurus, but they also said that they were beginning to use Montdorensis on hydrangea crops in the Netherlands with some success. So I suggested to our growers that they switch from Cocumurus to Montdorensis, and within quite a short space of time, they were getting much better control. So we have got better prospects for control of Cytosis now. We still don't know why Cocumurus might not be eating them. It could be because Cytosis larvae are bigger than those of western flower thrips. It would be quite interesting to do an experiment on that. Some of you may have heard or even used food supplements to boost numbers of predatory mites, and there's various formulations of these. BioBest have a pollen product called Nutrimite, and BioLine and Coppet have products that contain prey mites. And they're recommended to be most useful when there's low prey densities or when the plants are not producing pollen, because all these predatory mites feed on pollen as well as thrips. What I would say is that most of the research and commercial use of these products has been overseas on protected edible and cut flower crops, and haven't really done any research over here on bedding and pot plants. So it would be quite good to do that. So if BPOA would be interested in that, to see how what are the cost benefits in using this for either thrips or whitefly control, or would it be as good just to stick a few more predators in, then we could talk about doing that. So Alicia's already talked about our microbial entomopathogenic fungi. For thrips, Naturalis, Botanigard and Mycotel, which is a different fungus than Naturalis and Botanigard. They're all approved and on the label for whitefly, but they'll also give some control of thrips. But being a fungus, they need warm, humid conditions to work. So it's best to apply them on humid evenings or on pot mums just before blackout. But don't expect a quick knockdown. The fungus has got to germinate and grow and then kill the thrips. I'd just like to show you a quick result of a trial we did in the MOPS program, where we tested using Botanigard tank mixed with Majestic as a supplement to Cocumurus. So this is our water control with lots of thrips larvae. This is our Cocumurus, which gave pretty good control, but a tank mix of Botanigard and Majestic was even better. So it showed you can top up your Cocumurus or your predatory mites with a fungus to help out. And that was actually as effective as topping up with Actara, which is no longer available, but we used it in the trial because it used to be pretty good against Western flower thrips. So that shows that it can be useful. And just to give you a taste of commercial use of Botanigard and Majestic against thrips, we did a trial in the Amber project at HH where we compared weekly applications of Botanigard tank mix with Majestic with their regular Nemesis nematode program. And we used them in the last 3 weeks of the bud and flowering stage, just before marketing. And both treatments kept thrips very low numbers, less than 1 per 20 pots. So at low densities, Botanigard tank mix with Majestic was as good as Nemesis. And those of you who saw the BPOA webinar last October, where Marius from HH joined me in talking about bioprotectants for thrips control, will remember that he gave details of their IPM program on pot mums, where they use a combination of predatory mites, nematodes, and having seen how effective Botanigard could be in the Amber project, they're now alternating Botanigard and Naturalis tank mix with nematodes in their IPM program. And they're finding that this gives good control when thrips numbers are low or moderate. I'll just mention quickly a bit later what they do when thrips build up. So they put both the nematodes and the fungi on using their overhead spray boom just before blackout to maintain the humidity on the plants overnight. So just a very quick, I'm nearly at the end now. Azotin, Alicia's already just mentioned that. So this is a natural substance bioprotectant. It's an extract from the neem tree. We included this in our Scepter Plus trial in 2018 against Western flower thrips on verbena. It was actually the most effective treatment, gave over 90% reduction of thrips and 80% reduction in flower damage. So it was very effective, and it is IPM friendly. Going back to HH, they often find that thrips numbers build up in the summer in August and September. So last year, for the first time, they tried Azotin. So instead of using Botanigard or Naturalis tank mix with the nematodes, they stuck Azotin in instead, and they found it was very good and gave better, integrated with their biocontrols better than Conserve. So if you haven't tried it yet, it's worth trying. And finally, 2 more natural substance bioprotectants, Majestic and Flipper. We included these in our verbena trial in the Scepter Plus. And I was actually quite surprised how well they did, really, because they're purely contact in action. And they also gave pretty good control of thrips and flower damage. But towards the end of the trial, when thrips numbers increased, both the Majestic and the Flipper lost control. So I would say don't rely on these if you've got a lot of thrips that you want to knock back. And just to clarify, there's a bit of confusion because there's various maltodextrin products on the market. Majestic and Eradicote are actually identical products, just in different packages. And in August of this year, there'll be a new product launched, Eradicote Max, which is a new formulation, which is claimed to be better against aphids. So that will replace Eradicote. We haven't tested it yet ourselves against thrips, but we have tested it against mysis and on hebe and leafhopper and sage, and gave useful control. So that's one to watch out for. I think I've just mentioned Flipper. It actually gave similar control in our verbena trial against Western flower thrips, similar control to Majestic. But again, when thrips numbers increased towards the end of the trial, it ran out. So again, don't rely on it at high thrips densities. Finally, I'd just like to mention a new thrips trial we've got this year. AHDB wanted some work done on other thrip species on cut flowers. So this is going to be led by Andy Gladman. I'd like to introduce Andy, our new horticultural entomologist. He's based at Boxworth near Cambridge with myself and Alicia. He's going to manage this trial. We're using pot mums as a model crop. We'll be testing novel bioprotectants and conventional insecticides. We'll be building an R-Septor Plus trials on Western flower thrips and work on onion thrips that have been done on leek at Warwick University. So hopefully that'll come up with some new information that's useful for you. If there are any cut flower growers out there who've had any thrips problems, if you could get in contact with Andy, I know he'd be pleased to hear from you. So I'd just like to thank everybody who helped me put together this presentation, and to remind you of the fact sheet where all the information is there in full. And I don't know if we've got any time for any questions, Chris.
Well, just a quickie. In the chat, we've put a link to, or Alicia's put a link to the Thrips poster, which is very good. So everyone can find that if they go into the chat. A couple of quick questions from the chat, really sort of quite simple ones, but probably quite interesting. So first of all, do thrips leave cast skins that you can see?
They do. The only time I've really been aware of them is when we've done studies with predators against thrips, and what they do is suck out the body and they leave the little skin behind. But because there's only two larval stages of thrips, the first and second, and the first larva is very small and transparent. So unlike aphids that have got more immature stages, you get more cast skins. The thrips, there's very few, and they're very difficult to spot, really.
Okay, thank you very much. Okay, and then we've just got the poll has just come up. The last one. Would you like to everyone quickly like to complete the poll? And I'll just ask one more question of Jude while you're doing that. A question was also asked about the use of traps outside to trap thrips. Is that useful?
Yeah, well, I know that in onion and leek, traps are used for onion thrips, but outside you get a hell of a lot of all sorts of other insects on, and also a lot of soil debris that's blown around by the wind and things. So you can use them outside, but I wouldn't use them outside for long because they'll just accumulate all sorts of other stuff, really, and you won't be able to see any of the thrips.
Thanks very much for that. Coral, would you quickly like to put up the poll results for us? That's a resounding success, Jude. 100% for people who learned something new today.
Good, thank you.
So
trouble connecting to the internet. Check your Wi-Fi network connection by going to settings.
Thank you, Jude. And if David, you'd like to share your screen. Okay, David, we can't actually hear you at the moment.
Okay, so hopefully everyone can see that shared screen.
Yeah, you just need to be in presenter mode. That's all, David.
Okay.
Just straight up on the top. Just a little, up a bit. Up in the top ribbon, and the little. That's it. That's the one.
Right. Okay. Great. Right. Thanks very much.
Coral, if you actually work, David, you just need to go back on it.
So, got a poll here. Couple of images there, and which pests are responsible for this? The images you're seeing here. So, if you can all have a go at that, that'd be great.
David, you just need to roll back over that presenter button. You just rolled over it rather than clicked it. The one you just went over.
Is that working now?
Let's just got a bit of a delay. It's not actually happened.
Strange. Any good? If not, Coral, if you could share it, and I'll just tell you when I want.
David, you just need to put it onto slideshow.
That's the way.
There we go. That'll do.
Okay, great.
Perfect.
So the polls come up. We're asking who's responsible for the images here, and it's a bit of a tricky image, the one on the right, but I'll give you a clue. They are actually eggs. So, okay, brilliant. So 100%, everyone's got it. So that's great. So the image on the left is sooty mold. Image on the right, glasshouse whitefly eggs, and they are initially white when they're freshly laid, but they turn a browny black color before they hatch. Something strange going on here, Chris. It won't let me move on to the next slide.
David, do you want me to share my screen?
Yes, please. Yeah, if I just say next slide when I don't know what's happened. So if you can share and go on to the second slide, that's great, Coral. Thank you. Okay, so moving on to the whitefly life stages, it's useful to have knowledge of a pest's biology. Often products target a specific stage of a life cycle, so understanding that is useful and helps with your monitoring to link to how effective that intervention or control has been, whether that be a biological bioprotectant or a chemical control. So we've got the eggs on the left, the 4 larval stages and the pupal stage, which happens during the 4th larval stage, and obviously the adult, which you'll all be familiar with. This is glasshouse whitefly here. So next slide, please. So Bemisia tobacci, dreaded tobacco whitefly, its common name. Major problem comes in typically annually on poinsettia cuttings, affecting some growers in the country most years. Major transmitter of viruses, and that's one of the main problems associated with it, not necessarily for our crops, but very much for the protected edible sector. So that's why there's the strong desire to keep it out of the country and stop it establishing, because of the massive impacts it can have on yields of edible crops. So next slide, please. So I put this slide together, just hopefully a fairly simple slide, as an aid memoir to help identify some of the key differences between glasshouse whitefly, which can be common on nurseries, and tobacco whitefly. So just talking through the points, eggs of glasshouse whitefly, as I said, start off white and then darken, whereas the tobacco whitefly is yellower. The pupae for glasshouse whitefly are really quite oval. They're white in colour with very noticeable vertical slides, and You can see wax threads, but they can be a bit more difficult to spot, even with a good hand lens in the field. But comparing the pupae of the tobacco whitefly, they're flatter, they haven't got that steep vertical side, and you can sometimes see the red eyes of the adult depending on the age of the scale, the pupa. The adults, glasshouse whitefly, are bigger, and their wings cover the body, as the image on the left, which is a glasshouse whitefly. The tobacco whitefly image on the right, they're a bit smaller, they're yellower, and that's because they have less wax, and the wax gives the glasshouse whitefly its whitish color. And the wings are held quite tent-like, so like in that image, you can actually see the body visibly through the wings. And then just some notes on habit. Glasshouse whitefly flies up readily when it's disturbed, as I'm sure many of you have encountered over the years. But I've got less experience with tobacco whitefly because obviously it's a notifiable pest, and where you do see it, you don't see it in as higher numbers. But it's something I've noticed, and growers have told me they've noticed, that it does seem more stealthy. It doesn't seem to fly up into the air to the same extent as glasshouse whitefly. When it's disturbed, it sort of is keen to seek cover, it seems, and maybe that's partly because there are less of them when you're disturbing them, but that's just a tip that might help you. So hopefully that's a useful slide for people to refer back to if they relook at this presentation in future. Okay, next slide, please. So we've covered a couple of the important species, glasshouse whitefly and tobacco whitefly. We've also got the cabbage whitefly, sometimes referred to in the industry as a brassica whitefly, and it's got these fairly distinctive black spots on its wings, 2 on each wing. And actually some growers say this can be more troublesome, particularly on poinsettias, than the 2 species of whiteflies we've just been talking about. And that's because it is capable of breeding on poinsettia, despite poinsettia not being in the brassica genus. Also, it's highly visible in that it leaves a waxy deposit on the leaf, sort of merely waxy deposit, and where it lays its eggs, and it is capable of breeding on the crop. There are some challenges with biocontrol of this pest, and I'll revisit that more when we get into the biological control section of my presentation. So it obviously can migrate in and from brassica crops and oilseed rape. So that's that one. Next slide, please. Okay, so cultural steps to control and prevent whitefly, the same as from many other pests, really. Cultural controls are key, and monitoring, and particularly monitoring incoming stock, particularly if you're clear of whitefly, really is worth looking at bought in young plants and having a good scout through those, because there may have been a control applied for whitefly which might have killed the adults, but there might be some scales there that are waiting to hatch. And if you pick those up as part of your sort of incoming plant check, if you like, that stands you in much better stead for being ahead of a pest before it spreads onto other crops and associated impacts. So biological control can be a little bit more challenging on bedding type crops, perhaps more so than pot plants because of the short nature of bedding crops, but that's something we can revisit later in the presentation, and we'll cover the options you've got for control. Also, there's a link there to the whitefly fact sheet, which is available through AHDB archive site, and there's also a link to a webinar on managing Burmesia and poinsettia for poinsettia growers amongst you. Okay, next slide, please. So cultural controls, focusing on that a bit. So weed control, very good method of helping to prevent that green bridge, particularly at the end of season cleanup, getting the nursery nice and clean, preventing that pest carryover. Hygiene important, basic stuff, just a reminder of it really, but also in the thrust of the season, there's a need to manage and control populations, and that's however you do that, whether that's chemically or biologically. But being aware of the population that you've got on the nursery, whether that's on incoming plant material or within the nursery, and managing that to keep it in check and prevent it causing problems. And also not being tempted to think those plants in the corners are ready for disposal. We don't need to look after those. We're flat out busy. We haven't got time to throw them away, because they can become a breeding ground for pests such as whitefly and other pests covered today, particularly the mobile ones such as glasshouse whitefly and thrips, which can then spread when you dispose of that crop onto other younger crops that are still saleable. So, and also I have had people that have had a batch of stock with a burden of whitefly, and they have actually sprayed it, even though they're going to dispose of it, to prevent that spread, given the mobile nature of the pest when the crop's disturbed for disposal. Also, you'll see here the yellow roller sticky traps that help to catch adults and control spread within the crop. And a monocrop here, so poinsettia, but where you've got crops, different more bedding type crops in the season, different batches of stock coming perhaps from different suppliers, the sticky yellow roller sticky traps can help prevent movement within the glasshouse. Also, you'll see red circle around there. That's an aubergine trap plant that some people use to help monitor and draw whitefly to them. Just a sort of comment really about trap plants. They're fine and good if they're well managed, but you need to be aware of the pests that they can get and not let them become a breeding ground for pests. And also hyperparasites of aphids can potentially build up on some banker type plants, so just be aware of that. Okay, next slide please. So carrying on with the monitoring of whitefly, as I've said, really checking plants thoroughly when they come in is very good. Having some sort of plant health check rather than just merging them straight into production, and that can really help to prevent problems spreading. We talked about indicator plants, and yellow sticky traps are best for whitefly. You will catch some on blue traps, but yellow are considered best. Very useful guide to help monitor pest numbers, but they need to be changed. This is a nice new trap, but I try and encourage people to put the date that the trap was put up, because that also helps with changing traps. And then what you're seeing on the trap is useful. If the trap's put up in February, March, and it's still there in June, and it's really difficult to interpret what's on there. Also, this check type trap is useful if you're recording or doing any counts of whitefly that you're catching on a sort of weekly basis. So just write the date the trap was put up with a biro on the trap. That's what I'd encourage people to do. Also thinking about where traps, monitoring type traps are positioned within the crop. If they're 10 to 20 centimeters above the plant, you'll minimize your beneficial catch. So you will still catch a few beneficials, but you just have to accept that, frankly. Okay, next slide, please. So starting off with predatory mites for whitefly control, rather than parasitoids, because some of these are active at lower temperatures than the parasitoids. So we've got three main options here. They're all for use in permanent protection with full enclosure, but well worth checking with the supplier before use in tunnels, as that can vary in different countries. So for some products, it's different in countries such as Wales, if we've got any Welsh viewers with us today. So got the three predatory species of mites there. Something strange going on, Coral. I'm seeing an email appearing. Thank you. Three main species of mites there. So they have a temperature range, quite a wide temperature range they're active at, but most are said to be optimum at about 20, and Swirsky is particularly a warmer species that needs higher temperatures, but they are active at lower temperatures. But as a general rule of thumb, where there's no heat in sort of bedding crops or minimal heat frost protection, introducing predators from early April, temperatures are normally sufficiently high. Something else strange going on the screen here.
I do apologise, sorry, something's going on with my computer. I don't know what's happened, sorry. That should be good.
Okay, yeah, it's gone again now, thank you. So also these species, as Jude said, will contribute to thrips control, so another win there, particularly if you've got crops that get thrips, as well as whitefly, which many do. Just a point about Lemonicus, that there is an upper temperature limit in the literature, but certainly some of my growers that use it throughout the summer under glass, albeit in modern, well-vented high houses, they don't seem to see any reduction in efficacy with this predator in the heat waves and blasts of heat that we seem to get over the summer in certain weeks. So that doesn't seem to be a problem for them, and they have very good results with the predator. So just wanted to make that point, really. Okay, moving on, next slide, please. So on to parasitoids for whitefly. Got two options: good old Incarcia formosa, tried and tested, been used for a long time. I think it was one of their first biocontrols to be commercialized, but it's more effective against the glasshouse whitefly, where it'll give you a black scale, as in the pictures on the right. It will attack Bemisia tobacco whitefly, but you get a brown parasitized scale. Also, just referring back to the foe I mentioned in one of my earlier slides, the cabbage whitefly, Incarcia will parasitize that, but it can't be relied upon as a control. So if you get that, you need to do other things. Some growers have reported some success with Chrysoperla, and that's of interest. Perhaps now the cost seems to be coming down a little bit, but if you get a high influx, you are likely to need to revert to a systemic insecticide for control of cabbage or brassica whitefly in poinsettias. So Erecmaceus is the other option, more effective against Bemisia, where you get yellow parasitized scales. It also tolerates higher temperatures, which is useful. And also just making the point that Erecmaceus can actually doesn't parasitize the cabbage or brassica whitefly at all, so it has no effect. So just wanted to make that point. Both of the species are supplied as a mix, and they're a very good option for crops such as poinsettia, where you potentially will encounter both of these whitefly species. Next slide, please. So just a few tips for success with parasitoids for whitefly control. Using them preventatively, like many biocontrols, when temperatures are sufficiently high enough. They will fly at slightly lower temperatures than 18, but 18 is a good round temperature for things to be effective, and days like today are below 18, but in ambient crops, I'd still be putting them in at this time of year where you've got a whitefly risk or whitefly population, because hopefully we don't get many days below 18 degrees at this time of year under glass and tunnels. Also interesting to note that the presence of honeydew can deter egg laying. So picture on the right is an arbutylon. So if you've got a level of infestation as that. I wouldn't start from here with Incarcia. It would be more appropriate to use some knockdown sprays to get the population down, and you could use physical products such as Majestic Eradicote for that purpose to knock the numbers down before going with your Incarcia.
Can I just butt in, David? Just to say we're getting a little short on time.
Yeah, okay, I'll keep pushing on. With your cards that your scales on, keep them out of facing away from direct sunlight. With any predator, weekly monitoring is absolutely essential, so you can track how the pest and predator ratios are going and whether you need to up your rate of predators or make any other interventions. I've put the links to a side effect databases further on in the presentation, so they're there for you. Next slide, please. So just a few points about microbial bioprotectants, entomopathic fungi for whitefly control. Jude's talked about the products for thrips control, so I won't labour the point on those too much. But also, really, I just wanted to flag up the temperature and humidity requirements, and these are best applied on humid evenings. Well, that doesn't fit the general nursery's working pattern, particularly in high summer, where people tend to start earlier in the morning and finish sort of early to mid-afternoon. So if you're using these products, do bear in mind the temperature and humidity requirements, because they're key to success, but not necessarily in high summer, but other times of the year. You can think about manipulating the environment, the growing environment, to optimize the efficacy of these types of products. So if you think humidity is not going to be high enough, there are techniques such as damping down path. It's another job, but if you want to make these products work, it can. Also controlling the venting to build a bit of temperature and humidity if appropriate. But there's a lot to think about in that area, and the key thing to have in your mind is, if I do this to make these sorts of products work, am I increasing my disease pressure and disease risk? So that will be very much dependent on the crop you're building. But I'll just encourage people with new builds, new glasshouse structures, to really seriously think about automated spray application systems, because we know we've lost a lot of insecticides. That's a trend that's not going to change, and we're becoming more reliant on softer, more natural type controls. And if we've got automated systems that we can set automatically to apply these types of products, even in high summer at the right time, they can still contribute to our control and keep us producing good crops. Next slide, please. So just listed the bioprotectants really for use in IPM, just as a reference to look back on, and the number per crop. So next slide, please. Okay, so obviously there'll be a need at times to integrate conventional pesticides alongside your biocontrols, including your bioprotectants, and some products there, and just sort of categorize those. You'll be familiar with many, if not all of them really. And just making the point that you can look at some tank mixes where you need to have a quicker result than perhaps a product like Main Man would have on its own. So tank mixing it with Majestic or Eradicote is an option. Okay, next slide, please. And these are the main conventional pesticides for use in IPM. Just to put those up with the sorry, I made a mistake there on the slide. It should be IRAC codes, not FRAC codes on the right, but they are the IRAC codes. It's just the wrong title at the top of the screen, so apologies for that. And it's important to keep alternating your IRAC codes to prevent resistance developing and building up if you're using conventional products. But bioprotectants and biocontrols will help with that as well, so you're minimizing pest population exposed to insecticides. Okay, next slide, please. Just thinking about spray application and quality, it's not something that always gets considered as it should really. So just wanted to make the point, medium spray quality is useful where there's no other advice, but fine is quite a good option for contact acting products as a general comment, but refer and read to product labels for further guidance. Next slide, please. Just touching on water volumes, because it's an area of interest. Many products say good coverage, and some labels recommend water volumes in the regions there, some which are obviously on the high side. Not all labels recommend water volumes, some do. Some recommend them at higher water volumes and perhaps the operator may like to use, but where there's no recommendation, it's left to the operator. But the main message being, if water volume's too high, much of it will end up on the floor, not the crop. So the lower the water volume, generally, the better the retention on the crop. Because what happens as you put a higher volume on, the water droplets or spray droplets coalesce under the influence of gravity, and then they start to drop off. So spraying to a point of runoff is not really best practice. Next slide, please. So this was looked at a bit in the Amber project, and various different water volumes were tested, and the amount retained on the plants recorded, and just the main message there: lower water volumes give more deposition, reduce waste and labour time for application. And because water's on tap in ornamentals, people tend not to think about the time and cost for stopping to fill up, but it all adds to costs. But you need to stick with the label, even if you are looking to reduce water volumes, and not contravene the label, because what's on there is a legal requirement. Okay, next slide. I'm nearly finished, Chris. Other products have a rate per mil or litre, so that's different for different products. They tend to be the more horticultural products and products we're using under EAMUs, but on pot chrysanthemums in the Amber project, maximum retained was at 1,000 litres a hectare, but smaller volumes on smaller plants. Right, okay, that's that. Next slide, please. So this is just my slide with the links to the side effects manual, and a couple of them already been mentioned, but you can download the Bioline Agrisciences one onto your mobile phone. That's quite useful as well. Okay, I see there's quite a bit going on in the chat, so I'm just going to tickle through that in a minute. Thanks to Russ Woodcock in particular for sharing some of his experiences to help put this presentation together. Right.
Thank you very much, David. Yes, as David mentioned, there's a bit going on in the chat. So if you just want to have a look at that, discussion about parasitoids and hyperparasites. If you'd just like to, I think we need that. I think we need the next poll, don't we, Claire? Coral, sorry. Have we got the last poll?
I might just have to wait a second. Sorry, I've got to shut down my screens.
That's fine. I think we could. I think we're quite happy that we've all learned something. So let me just thank everyone for their contributions. Thanks to Alicia and Jude and David. Obviously, there are the recording will be available, and then obviously links and things in the chats for bits like the Thrips poster. Thanks for all for turning up. Well, obviously at a busy time of year, and thanks again for everyone, and thanks to Coral for organising. Okay, thanks very much, and goodbye.
Thank you. Bye.
Bye.